Hi, my name is Preeti Bais, and I work for the Jackson Lab for the Genomics Medicine as a statistician and bioinformatician. I got the joke. With a name like Preeti Bais, working as a statistician is such a hard job, you know. You never get, yeah. And before that, I worked for a small startup company, and whenever they introduced me to one of their clients, they were like, every client went like this, you know. They have a pretty biased person as a statistician. So here we go. So today I'm going to talk about one of my older projects that I did during my PhD. And the reason being, I'm going to talk more about the methods. So I took those plant data as an example, because first it's very easy to manipulate the plants, and you can grow them in controlled conditions. That's what the statisticians like. And the other thing is, I could use that data without, upsetting anybody else. So here we go. So in my PhD, what we did was we took Arabidopsis as our model system, and we used metabolomics to understand the functions of the genes. Why do we need to do that? Because there are about 27,000 Arabidopsis protein coding genes which have unknown functions. 9,000 of those genes either share no sequence homology to any gene in sequence databases or share homology to genes of unknown functions. So you can't find the function of those genes just by doing the sequence analysis. And 15,000 of those 20,000 have, you can only assign function using a broad category like phosphatase, kinase, etc. So then we introduced metabolomics to the system. And what we did was we had 140 single gene knockout mutants, and we created a database which is publicly available. This is www.plantmetabolomics.org. And it not only contains the data for those 140 single gene knockout mutants, but it also has software for all kinds of data analysis. So yesterday I was talking at lunchtime, and people were complaining that there is no software that anybody can use and analyze their data. So this software is publicly available. It can do your basic analysis like PCA and stuff, and also do random forest and all that Dr. Beecher was talking about yesterday. So the idea is, if an enzyme works between metabolite M and metabolite N, and if we knock that gene out that codes for this enzyme, then we should see metabolite M going up and metabolite N going down, and everything that follows N downstream of N going down-regulated. Okay? So this is an example of what we have in the database out of the 140 mutants. So I'll discuss 3 mutants in this talk. And we focused on the GSH degradation pathway. And we took 3 mutants, 5-opase and 2 mutants of GGT. So 5-opase works between 5-op and converts 5-op to glutamine. And GGT converts GSH to gamma gluamino acid. I don't understand all that. It's just difficult names. So just remember that this works between these two. This works between these two. And this is mammalian pathway. So there is a gene, single gene, 85G37830 in Arabidopsis, which has very high sequence homology to the mammalian gene. This one, okay? And we got this gene from this group, which had knocked it down, and we put it in our system. So when we looked at the biopsych pathway from Dr. Karp's group, we wanted to see what do they say about this pathway in Arabidopsis. So for biopsych is a good, very good source for all the predicted pathways, and they also have experimental and literature in that database. So for example, if something is predicted, so this is GGT1 and GGT2. So they specifically said that there is no experimental data which confirms this. But for OXP1, they had experimental data, and that's why they put it as bold. So this is a very good, quick and easy source for finding out if there is an experimental data for the pathway that we are looking at. So these were the genes that we were looking at. And the experimental data they had in biopsych is the same group that donated us the mutants, all these 3 mutants. So this was our experimental design. So Dr. Suri, who is also a collaborator of Dr. Karp, chose the 140 mutants for us that we wanted to analyze. And we took those mutants, grew them in controlled conditions, and then allocated them and distributed them to 6 labs, which used 11 different metabolic platforms on the same biological material to generate the data for us. So we collaborated with Dr. Feen and several others, okay? And in the end, we had about data for about 1,500 metabolites, out of which 50% were unknown. So this you can think of as, there were 10 PIs and there were only 2 students. So you can think of that as an like upside down cone, but it worked, okay? So okay. And this is the data analysis pipeline, and everything in this pipeline is already available as a software in our website. So you can run this pipeline using just some clicks of the buttons. So we do replicate quality analysis within a platform. So within a platform, for example, Dr. Feen's platform will do, if he was using 6 replicates, we would see the variation amongst those replicates. Exploratory analysis, so all PCA, all ratio plots and all those fold change analysis would come here. And then normalization, so we use best practices and we give options, different options that they want to use. And the help is very good as well. So it actually tells you and guides you what normalization you should use. And then the idea is to compare mutant with the wild type control, okay? If the samples are separable using random forest method, then the next step is to find the important metabolites. If they are not separable, then we can hypothesize that the mutation may not affect that metabolite family, okay? And we can hypothesize the gene function. But if it's separable, then we look into the metabolites and find if they are structurally known or unknown. And as Dr. Beecher was saying, about 50% of the metabolites are unknown in any system. So then we proposed a correlation analysis based method to find the structure of the structurally unknown metabolites for free, actually. And then if they are structurally known, you can put them in pathways. One of them is Dr. Karp's pathway, and see, hypothesize the gene function. So this is the pipeline, and it's all available as a software, and you can use it. So this is the database, plant metabolomics database. We not only have the data, pictures, but we also have all these tools that I was talking about. This is the correlation tool, the QC tool that I was talking about here. So all these tools are available, and then we have connections to different publicly available databases. For example, BinBase is from Dr. Feen's lab, Plant Metabolomic Network is from Dr. Suri's lab, KEG, and then lipidomics pathways. So it's all connected to this website. And these are some of the examples from the software. So this is the OXP1, this guy here, which works between 5-OP and GLU. So this is the ratio plot of all the different platforms that we used. And we see that some of the metabolites are changing a lot, and most of them are not changing. So y-axis is just the metabolite number, and x-axis is the ratio between the mutant versus wild type. So 5-OP, which should have increased in the mutants, is actually increasing. And plus there are some other metabolites that are changing quite a lot. And most of the data which is changing is coming from the GCTOF platform from Dr. Fiends. Then this is an example of the PCA analysis. Again, the GCTOF was able to distinguish between the two types of samples, the wild type and the mutant, but the lipidomics platform wasn't able to distinguish between the two types of samples. So here we start formalizing, hypothesizing that maybe the GCTOF is the platform to go for this mutant. Okay, and there may not be any changes in the lipids for this mutant. Okay, and here is the random forest analysis for all the 3 mutants. So this is the OXP1 mutant where we didn't see anything in the lipids, but we saw something going on in the GCTOF. And this is the random forest that also could separate the wild type from the OXP. But none other platform could distinguish between the two types of samples. And actually the error rate was very similar to when we compared wild type with wild type. So whatever was there was not significant. And in the GGT1 and GGT2, we couldn't distinguish between the wild type and the mutant, which is not a bad thing, because this shows us something that maybe there is nothing changing in the lipids for that mutant. Maybe nothing is happening with these 2 mutants. So this also gives us some information about the mutants. So then we did the next step for the GCTOF and tried to find the most important metabolites for wild type and for the difference between wild type and the OXP mutant. And we found a list. But there was another problem. In this list, oxoproline is upregulated in the mutant, which is a good thing. GABA is downregulated, but then there were 6 of the metabolites that, or metabolic features that we didn't know anything about. So this is an unknown, and it's a very big problem in non-targeted metabolomics, that you see a lot of unknowns. So once you have unknowns, you can't put them in any pathways or do anything with them unless you do more experiment. And in the bottom, I'm showing how these are changing. So oxoproline is highly upregulated, GABA is going down, and so on. And this all can be generated using the software. Okay, so we were kind of stuck here, and I wanted to graduate. So my professor said, we don't have any money left. We used up all our money generating the data. So you come up with something that is inexpensive, in other words, free, that can solve our problem, okay? So here I am. So I started looking at the literature. And I thought, why can't we do correlations in metabolomics? So I hit a roadblock again. People have been trying to find the correlations in metabolomics, and it never works. So they have not been able to do the topologies, biochemical topologies using the correlation. First of all, you get a hairball when you do basic Pearson correlation or a Spearman correlation. And the reason being, if 2 metabolites have the same diurnal changes, you will see them as a positive correlation. And if they go in the opposite direction, just the way they fluctuate, then you will see negative correlation, and it's not a good thing. What you want is they are in a pathway, and when you block that pathway, they are going in opposite directions. So this is what you want to see. This you don't want to see, but you get a lot of this. So correlations are very high in large scale omics data set. So people suggested using, and this is a big group who is working in correlations, okay? So they suggested using partial correlations, and I'll tell you what a partial correlation is later. And then they also suggested using many mutants or many different conditions to see the correlations. And if it can stand the test of different mutations and different conditions, then that correlation might be a real correlation, not artifact. So we were lucky because we already had data for 140 single gene knockout mutations. And all I needed to do was do partial correlations. So this is what a partial correlation. Again, my professor always said that to make yourself look important, put a slide that nobody can understand. So here is that slide. So partial correlation is between, if you're looking for the partial correlation between 2 points, point 1 and point 2, you want to remove everything that is correlated because of their correlation with a third point. So what you're trying to do is you're trying to remove the effect of all the other metabolites on the 2 metabolites for which you are trying to look at the correlation. And this gives you a sparser, why am I here? This gives you a sparser hairball. You always get a hairball, but this one is sparser. So, and then this paper came out in 2011, which showed the first order partial correlation, Gaussian graphical models, GGMs. And they showed, I won't go through all the examples, but here is an example. So this is a network, A to B to C. And if you do a normal, regular correlation, Pearson or Spearman, you would see that all of them are correlated to each other, which you don't want. You want A to B to C. But when they did the GGM, they did A to B. They actually could mimic the topology of the real network. And they did with, their data was not real. So they generated this data and they showed different kinds of topologies. So we used their paper and thought, let's run a first order partial correlation and see what happens. So, what we were trying was to find the neighbors of interesting metabolites in the correlation networks and use intuitively identifying the unknown metabolites. So here is the list again, which I showed you earlier with 6 metabolic features which were unknown. So out of the 6, we found highly correlated metabolites for 3, so 50% success. But still, it's better than knowing nothing about those metabolites. So this one, 200489, is highly correlated with 5-OP, which was going up. So, and again, so we found 3, and they were all correlated with 5-OP, which was interesting, and they were all going up in the same direction as 5-OP was. So this is where I'm trying to explain why all the amino acids are going down and why, and as biochemists you already may know this. So in summary, we generated a database of 140 single gene knockout mutants, and we used 7 to 11 different metabolic platforms for every mutant. So we have data for 1,400 metabolic features on each of these mutants. So this is one of the most comprehensive databases in experimental databases in plants for Arabidopsis, and it also has the online software tools for data analysis. And then finally, there is an inexpensive method to understand the important features of unknown structures. There are some caveats, but still. This is the group. This is the upside down cone I was talking about. So all the PIs and plus the 2 students. So this is 1 student and I was the 2nd student. So she did all the plants, and she was a plant biologist, so she did all the plants. And I'll leave you with this. We are looking for, in Jackson Lab, we are looking for mass spectrometry scientists and looking for collaborations as well from the PIs. So if I can hook you up with the PI, big people in Jackson Labs, but you have to promise me that you'll give me the data to analyze. And that's it. Thank you.
Suggestions. Well, the first suggestion is when you're working with the different laboratories, having different platforms in metabolomics, the first thing I would do I would send them QC. And
Yeah.
they run the QC, they give me back the results. When the variability of the results acceptable, then I would send the sample.
Yeah, so we did, so what we did was we sent them the same material, and we ran a lot of QC experiments before also.
No. You have to send them QC without telling them that it's QC.
Oh, okay.
They will get you back the numbers, the measurements.
Sure.
If those numbers for what's inside the standard QC
Yeah.
have to be the same from the same platform,
Yeah.
or within the, 20% variability.
Yeah.
That the first step before you send the real samples,
Yeah.
right? When you send the real samples and you get 7 laboratories found nothing and 1 laboratory found something, that QC may help you to alleviate this maybe discrepancy, maybe not.
Yeah.
Why is one lab is working much better than the other labs?
Yeah.
Because it's very simple. All the data from CEMS, LCMS, and GCMS has significant overlap in amino acids and fatty acids. That should be your QC beforehand.
Yeah. So on that comment, so for that particular mutant where I showed.
Yeah, I know all these guys. Yeah.
Yeah, so only DC top was non-targeted. Others were targeted. So
Definitely.
We were using like these 7 labs and all these platforms, but we actually didn't use all of them in all the mutants. So it was.
Sure.
So,
yeah.
so
I understand. I just go back to principle. I'm not criticizing what you've done.
No. Good suggestion. Yeah.
Yeah, and yeah, you wanted to say something, right?
Yeah. My impression is if you look at the list of compounds, they have been chosen to be complementary, not to handle the same things. That's what you can see from the data.
If before you submit experiment, you validate the platform.
Of course. But then depends on what you can check.
If they come up with the different things, it has no value.
No, they measure different metabolites.
What I'm trying to tell you that each of this platform has overlap. Each of this platform that's used can measure amino acid. That should be your QC before. Well,
Yeah, of course.
maybe not. Sorry. Yeah, I'm sorry. Probably ran
Yeah.
out of time. Thank you.
Thank you.